All right, everyone, welcome back. I think we'll make a start. Okay, so this week we're continuing on looking at cytotoxic T cells and lymphocyte development. But today our focus is going to be on B and T cell development, as well as looking at immunological tolerance and how that is mediated. So these are the learning outcomes for the lecture today. So you'll be able to understand the key steps in both B cell development, but also T cell development. You'll also be able to gain an understanding of what positive and negative selection means, as well as immunological tolerance. And then you'll also be able to compare and contrast central and peripheral tolerance. And then also have a quick look into how tolerance can be lost and the specific mechanisms that can actually sort of lead on to autoimmunity. And we'll just have a quick look at rheumatoid arthritis. So the attendance pin for today is 8791. So that's 8791. So this is obviously just a little bit of a recap. You've obviously seen this before. This is hematopoiesis. And so just to re-jog your memory, that obviously when it comes to lymphocytes, they're derived from the common lymphoid progenitor. And this is what is sort of delivering our adaptive immune response, this right-hand side of this diagram here. Now obviously I want you to bear in mind that the bone marrow is a very dynamic and complex structure. And this is what is containing your hematopoietic stem cells that obviously give rise to that lymphoid lineage. And this commitment to either a B cell or a T cell lineage is really sort of regulated and controlled by a whole range of transcription factors. But just remember that obviously the B cell development is taking place in the bone marrow, whereas the T cell development, or the progenitors, will begin in the bone marrow, so they'll rise in the bone marrow, but they'll finish off and complete their development within the thymus. And that's just what's being summarized in the diagram on the right-hand side. And it's this microenvironment that is provided in both the bone marrow, but also the thymus, that is really helping to support the growth, but also that lymphocyte development. So, you know, the microenvironment is playing a key role in the development of these cells. And the different stages and development of these cells through hematopoiesis, it can be quite easily defined by looking at specific cell surface markers, but also the expression of particular transcription factors. But in addition to that, the rearrangement of either the B cell or the T cell receptor. So looking at all of those can give you a good indication as to what stage the cells are at in their development. So if we look initially at B cell development, so this is not as complex as T cell development. So if you remember back to a couple of weeks ago when we were looking at this, hopefully you'll remember that B cell development will actually sort of begin with the rearrangement of the heavy chain locus. And then what happens is you'll get production of a pre-B cell receptor, and signaling through that will test to see whether you've got successful heavy chain rearrangement. And then what will happen, if that has occurred successfully, you'll get a transition from the pro-B cell to a pre-B cell stage, and they're just kind of listed in order down at the bottom here. When you get signaling through that pre-B cell receptor, that's going to inhibit any further heavy chain gene rearrangement, and this is what is actually enforcing your allelic exclusion. So remember we talked about that a few weeks ago as well. And these steps are all kind of dependent on signals from that microenvironment. So in this case, we've got our bone marrow stromal cells, and also you're getting regulation through particular transcription factors as well. So if we look at that in a little bit more detail, all the key steps. So essentially we've got a stem cell here, and then it's developing into an early pro-B cell and then a late pro-B cell. And it's just showing down here the rearrangement that may be going on with the heavy chain and the light chain, and whether it's expressing surface immunoglobulin. So when we're going through the B cell development, we're proceeding through these different stages, and we're going through the different gene rearrangements. So what you can see is in the early pro-B cell, we're rearranging our heavy chain genes. So initially we've got our D to J rearrangement, and then in the late pro-B cell, we've got our V to DJ rearrangement going on. you'll see that the light chain genes are still germline at this moment, and we're getting no expression of surface immunoglobulin. So once we've got a successfully rearranged heavy chain, we're going to sort of express the pre-B cell receptor on the surface here, and we're going to get signaling through Ig alpha and Ig beta as well, so remember those from a few weeks ago. And so what this will lead to is essentially the large pre-B cell, which can then proliferate into a small pre-B cell, which is kind of a resting cell. And when we're at this stage, we've got our fully rearranged heavy chain, but what we can start to do is we can re-express the RAG proteins, and that will start to rearrange our light chain genes. And so once we've got a successfully arranged light chain gene, this will become an immature B cell, and it will express IgM on the cell surface, and then later on, as it progresses through, we can obviously express IgD as well, and this is in conjunction with Ig alpha and Ig beta. So just to give a little bit of an overview of that, but in essence, once you have a productively rearranged immunoglobulin gene, it's going to be immediately expressed on that cell surface, on that developing B cell. The heavy chain gene rearrangement, it's initiated, if you remember a few weeks ago, by our D to J, and then we're getting our V to DJ arrangements that are occurring. If we don't get a functional heavy chain rearrangement that occurs, then that rearrangement can try again to see if we can get a successful rearrangement. Once we get that heavy chain being rearranged and the pre-B cell receptor is expressed, then it's obviously we get that signaling through Ig alpha and Ig beta, and that will essentially tell the cell, once we get that signal, that we don't need to have any more rearrangement, that we've got a successful heavy chain. If we can't produce a successful heavy chain, then we're going to end up with cell death. So remember that allelic exclusion where we had all that kind of cut-off points that we talked about a few weeks ago. So we have these large pre-B cells, they're going to stop dividing and they'll become this small pre-B cell where they undergo their light chain rearrangements. Remember back that we talked about the kappa light chains will be rearranged first. If that's not successful, we'll then get rearrangement of the lambda genes. And then once we get that successful light chain gene that has been rearranged, and we've got a complete IgM that's being expressed on the B cell surface together with Ig alpha and Ig beta, we can get signaling through that. And that signaling will stop any more light chain gene rearrangement. Likewise, if we fail to get a successfully rearranged light chain gene, we're going to end up with cell death. Okay, so we've got these checkpoints to make sure that everything is okay as we're going through these rearrangements. And then ultimately what will happen is that fresh B cell, we want to test to make sure that it's not autoreactive, and that will occur before it leaves the bone marrow. So how do we test for autoreactivity? Well, essentially what happens is we check to see whether that B cell or that B cell receptor can bind self molecules within the bone marrow. Now, if it does, that can lead to one of two things. We can have receptor editing, or we can have apoptosis and cell death. So on the left here, we have an immature B cell. It's not interacting or encountering any self-antigen. So what happens is it can migrate into the periphery and into the lymphoid tissues, where it will start to express IgD as well as IgM. However, if we have a B cell that is interacting with self-antigen, that's obviously a potential problem. So what happens is those cells can be eliminated by apoptosis, but they can also undergo receptor editing. So this allows the receptor a second chance, or the cell a second chance to kind of tweak its light chain genes. So if we look at receptor editing in a little bit more detail. So as I mentioned, this is essentially where we will replace our light chain genes to try and effectively rescue those self-reactive B cells by tweaking their antigen specificity. So in the diagram here, we have a B cell its antigen receptor is interacting strongly with our self-antigens, so we stop all of the development there. We don't want it to go any further. We start to re-express our RAG proteins. This enables it to undergo light chain gene rearrangement, and hopefully this will lead to a new productive rearrangement that will give us a new receptor. So this is the process that we would call receptor editing. So if the new receptor is not self-reactive and doesn't interact with self-proteins, then it's rescued. That cell is rescued and it can go on into the periphery. If, however, it still reacts with self-antigen, then it will undergo apoptosis. So it's kind of like a second chance, this receptor editing. So we either have receptor editing or death. Now, obviously, it's not going to be possible for all of the B cells to be checked to make sure that they're not self-reactive. Some are going to slip through the net, and that's where we can have checking going on within the periphery. So obviously, what was going on in the bone marrow there, that would be central tolerance, and this would be peripheral tolerance. So once the cells have left the bone marrow and they enter into the periphery and they're going through our circulation, they're known as transitional B cells, so they're not yet fully mature. But if these transitional B cells happen to bind a lot of self-antigens in the periphery, they will be eliminated. And so any transitional B cell that doesn't have a very strong self-reaction, it's permitted to actually continue its maturation into a mature B cell. And then during this actually takes place within the spleen, but then it will upregulate its surface IgD. So now you're getting a B cell that is actually expressing IgM and IgD in the periphery. So we have a look at T cell development. This is a little bit more complex, but remember that as we started off in the lecture, that all the T cell progenitors will arise within our bone marrow, but all the key stages of development will actually occur within the thymus. And like the B cells, the development of the thymocytes, you can characterize them by changes in not only the T cell receptor rearrangement, but also the expression of their cell surface markers and what signaling is going on. Now, it's worth mentioning that obviously the T cells, they're undergoing a huge amount of proliferation within the thymus, but actually the majority of these, about 95%, won't actually leave the thymus, and that's because it turns out to be sort of self-reactive. And if you look at this slide at the bottom, which is a cross-section through the thymus, you can see these red spots. They're actually T cells that are undergoing apoptosis, and you'll see that surrounding these, if you kind of have a little bit of a zoom in, you can see this blue staining, and they're actually macrophages that are essentially clearing up the debris from the apoptosis. So the thymus, you can see in this diagram here, it's this rather sort of, I don't know whether you'd describe it like butterfly-shaped, like bilobed organ, so it's sitting sort of high up in your chest. And it's worth mentioning that actually your thymus starts to decline in size after you've reached about the age of 20, and this is known as thymic involution. So from your early 20s onwards, your thymus is kind of decreasing in size. And the later stages of life, obviously that impacts your immune system as well, especially in the elderly. And that's a process known as thymic involution. So we start to look at the key steps. I just wanted to mention Notch signaling. I won't go into any detail, but it's kind of good to be aware of it. But essentially, the commitment to the T cell lineage within, it occurs in the thymus, but it is dependent on Notch signaling, so that plays an important role in kind of, how that T cell commits, what effector, function, or route it goes down. But essentially, the thymocyte development is quite tightly regulated by the rearrangement of the T cell receptor. So it's somewhat similar to B cell gene rearrangement. So it's got all these different stages here. And I just want to break them down into double negative, double positive, and single positive. And this is all classed on CD4 or CD8 expression. So we have our double negatives, which can be further broken down into DN1, DN2, DN3, and DN4. So I'd learned those names, the DN1 to 4. And this is based largely on the expression of CD44 or CD25. So you can see we've got CD44 positive, CD25 negative, and then we upregulate CD25, we then downregulate CD44, and then essentially DM4 is negative for CD44 and CD25. But none of these are expressing CD4 or CD8. When we get to the double positive stage, we're expressing both CD4 and CD8, and then the single positive is where we're either expressing CD4 or CD8, and we're kind of committing to whether we become a CD4 cell or a CD8 cell. And then underneath, it's just showing you the gene rearrangements. So I've just focused on the alpha and the beta genes. Obviously there will be something separate going on if we were looking at gamma delta T cells, but we're only focusing on alpha beta TCRs in this module. So you can see the kind of rearrangement in red and where it occurs during the stages of development. So what we have is initially we're undergoing this heavy chain, we're undergoing rearrangement of the beta chain. So we're getting our D to J rearrangement and then our V to DJ rearrangement. And once that occurs successfully, we're going to get the production of a pre-TCR, which you can see here, and we're going to get signaling through that, and that's going to prevent any more beta chain gene rearrangement. So we want to stop it once we've got a rearrangement that has been presented on the cell surface. Now, if that's okay, we can then undergo the VJ recombination of our alpha chain, and if this is successful, we'll then ultimately either get positive selection or apoptosis, and we'll cover these in the next few slides. And that's just showing you when they occur. So we're getting the beta chain rearrangements in the double negative stage, and then in the double positive we're getting the alpha chain gene rearrangements. As we mentioned, this is occurring in the thymus, and it's kind of occurring in different stages, and it's basically the cells are kind of moving between the medulla and the cortex of the thymus. So they're coming in through the venule here as DM1s, and they're interacting quite closely with the cortical epithelial cells. So these are the cells that you see here, and these are playing a really important role in terms of providing essential interactions with those T cells. So we move through the different stages until we've got our DM4 stage. We're then going to have our immature double positives, and then eventually when we come back into the medulla, we're going to start selecting for our single positives, whether we've got a CD4 or a CD8 T cell. So we have a little look now at positive and negative selection, because these play a really crucial role in this whole process. So when we've generated a T cell, we want to ensure that receptors, that we have cells surviving that have receptors that can interact moderately with self-peptide and self-MHC complexes. So we want to make sure that those cells survive. And this is known as positive selection, and actually plays a key role in selecting whether that cell becomes a CD4 or a CD8 T cell. And this is based on the specificity of the T cell receptor and how that interacts and binds with MHC. And it's these thymic cortical epithelial cells that we looked at in the previous slide that's actually helping to mediate that positive selection and playing that key role in developing or the thymocyte development. Any T cell, on the other hand, that's going to react very strongly with self-antigen is going to be deleted, and that's known as negative selection. So you want T cells that are encountering sufficient amounts of self-antigen in the periphery to be eliminated and inactivated, because obviously you don't want autoreactivity. And this is just another diagram that's showing you the different stages here from DM1 through to DM4, double positive and single positive. But it's showing you not only the gene rearrangements and the stages, but it's also showing you whether it's taking place in the cortex, the medulla, and where positive and negative selection occurs. So it's just nice to see it kind of laid out like that. So if we have a look at that in more detail. So this diagram here, it's showing you the thymocyte levels, but it's also showing you sort of the T cell receptor affinity for binding to self-MHC and self peptide. So we're getting stronger interactions on the right and weaker interactions on the left. So when we're getting all these TCR gene rearrangements occurring, obviously these are all random, we're going to get a huge amount of T-cell receptors, and inevitably we're going to get a lot of T-cell receptors that actually don't recognize anything, okay? And this is called no selection, so that's what this line is here. These T-cells, they're not going to interact with self-MHC or self-peptide. They're not going to get that signaling through their T-cell receptor that's going to provide them with survival signals. So they're going to essentially die by neglect, is what we call it. They're not going to get that survival signal. They're going to die, and that is called no selection. On the other hand, we're going to get some T cells that are going to be positively selected. So these are the T cells that can bind to self-peptide and self-MHC with a sufficient strength that gives them that survival signal. And that's what we want. So they're going to be positively selected, and they're going to survive. On the other hand, if we look at the far right of this diagram, we're going to have T cells that interact, or T cell receptors that are going to interact very strongly with self-peptide and self-MHC, and these are going to be negatively selected. And effectively, they're going to be clonally deleted, and they're going to be removed. So why is negative selection important? Can you think of a good reason why this needs to occur? What happens if negative selection didn't occur? Yeah, autoimmunity, because we would have T-cell receptors that are binding very strongly to self-peptide and self-antigen, and so that's a really bad thing. Now you've probably noticed in the middle we've got this little peak here which is called alternative selection. So this is a really small subset of T-cells. They're falling somewhere between positive selection and negative selection, but these will actually become our naturally occurring Tregs, okay? And they play an important role in regulating our immune response as well. And we'll come back to them in a few slides' time. So continuing to look at positive and negative selection. So positive selection is going to occur when we've got that T cell that is expressing the rearranged T cell receptor, and this is at the double positive stage. But then obviously we need to move into our single positive cells, so whether it's going to be CD4 or CD8. And as I mentioned, it's that positive selection that is helping to coordinate whether that cell goes down the CD4 or the CD8 route. But essentially what we're doing during positive selection is we're kind of testing, does this cell, does it fit better, you know, with MHC class 1? If it does, it's going to become a CD8. Or does it fit better perhaps with MHC class 2? In which case it's going to become a CD4 T cell. So they're just kind of testing to see whether a particular T cell receptor fits better with a certain MHC, and then that will dictate whether it becomes a single positive, it's like CD4 or CD8. So it's actually during that positive selection that we kind of determine whether the cell is going to become either a CD4 single positive or a CD8 single positive. And it's these thymic cortical epithelial cells that are helping to mediate that positive selection. And so obviously it's essentially requiring this cooperation between T-cell receptor, MHC, but also CD4 and CD8. So we've got that whole process going on, just determining whether that particular T-cell receptor would work better you know, on a CD4 cell or on a CD8 cell. So that's all occurring within the thymus, but essentially within the thymus we need to kind of test that T-cell receptor to determine whether, you know, it can bind to self-antigen in proteins that are perhaps normally expressed outside in the periphery. So, you know, how do we actually test for autoreactive T cells in the thymus when, you know, they're kind of within that microenvironment? So this is where the gene that's known as AIR is particularly useful. So this is expressed in the medulla of the thymus, and you can see on this slide here it's shown in green, and then the T cells are actually shown in red. So you can see that they're all kind of in close proximity, but essentially by expressing this particular gene, it allows the cells within the thymus to express proteins that would normally be found in the periphery. So this is part of central tolerance. So essentially within the thymus we're kind of presenting those T cells with proteins that would normally be found in the periphery and just seeing whether they auto-react. And again, any T cell that is going to bind to any of those self-antigens in a sufficient quantity is going to be eliminated or inactivated. And then likewise, if those T cells move off into the periphery and also encounter some perhaps self-antigen that they interact with, they'll be eliminated via peripheral tolerance. So we've got central tolerance occurring within the thymus, but we've got peripheral tolerance occurring outside, because obviously it's not possible to kind of test for, well, essentially some things will slip through the neck, so that's why we need peripheral tolerance as well as central tolerance. Now this is just a little diagram just kind of summarizing that. So we have immature T cells, they're interacting with those cortical epithelial thymic cells, and basically they're seeing whether they recognize self-MHC. Anything that recognizes or has a strong recognition is going to be removed and eliminated, die by apoptosis, and then that just takes you back to that slide again where in red we have all our apoptotic T cells, and they're being surrounded by macrophages that are actually removing that apoptotic debris. So this is just kind of that diagram again, showing you the cells progressing through their DM1, DM2, DM3 stage. And it's just really kind of showing some of the checkpoints. But obviously one of the checkpoints that we have here is checking whether they have, expressing a pre-TCR. That's obviously the first checkpoint. And then the second one would be checking that they actually have a successfully rearranged T cell receptor on the surface. And this is just another kind of summary of how the cells are moving between the cortex and the medulla, and where positive selection and negative selection is occurring. So you've already seen it kind of on that linear diagram. That might be more beneficial, or this might be more helpful just to kind of see it actually sort of within the structure of the thymus. But essentially they're undergoing positive selection in the cortex and then negative selection in the medulla, and then they're going off to enter the periphery and move into the circulation around the body. So if we just look at the importance of positive selection and negative selection, this really does play a very crucial role because it's shaping the T cells that you have within your body. And there's a couple of different criteria that, essentially we really need to stick to, because this is defining our T cell repertoire that we have for each of us. So the first criteria is that the T cells need to work effectively with MHC. So that's why we have that whole positive selection going on, to make sure that we have a good interaction with the T cell receptor and self-MHC, because otherwise it's going to be useless. So that's why we have the positive selection. The second criteria is that the T cells mustn't be activated by self-antigen. So that's why we have negative selection. So we need to, kind of select out for any lymphocytes that are bearing that high affinity T cell receptor, that can recognize self-MHC or self-peptide very tightly. So both of these together are really shaping the T cell receptors that you have within your body and how they interact with the MHC. So it's a very crucial process. As I mentioned before, obviously central and peripheral tolerance are really playing an important role within this whole process. Now, as you're well aware, it's the immune system that is responsible for defending your body against foreign substances or pathogens or perhaps even transformed malignant cells. And the key to enabling this is being able to distinguish self from non-self, okay? But also, whilst you're doing that, you need to avoid autoimmunity. So as you rightly said, we need to do this whole process in order to prevent autoimmunity and, recognizing our own self-antigen. And the body's ability to be able to recognize, but also tolerate self-antigen is what we call immunological tolerance. So this self-tolerance, is basically there's a variety of mechanisms that are going on. We've got the central tolerance, which we've been looking at, which is occurring in the bone marrow and the thymus. But we've also got peripheral tolerance, which is occurring in the periphery within that sort of pool of mature lymphocytes. Now, just to use this as an example, we have several immunologically privileged sites within the body. So, the eyes, the brain, the testes, also, the fetus during pregnancy. But these are immune-privileged sites, and they don't induce immune attack. But if you get damage to one of these areas, so let's say you have damage to one of your eyes, that's potentially going to release antigens from your eye into the surrounding tissues. This can make their way to the lymph node, and you can then start to sort of get activation of T cells, and these can then migrate into the healthy tissue of the eye and mediate an autoimmune reaction. So this is obviously not a good situation to be in, because this is leading to autoimmunity. So we need ways that we can kind of correct that, and that's where tolerance is coming in to this situation. So hopefully you'll remember Tregs, or regulatory T cells. So we talked about these when we were looking at the CD4 cells and the effector T cells. But also we were talking about cytokines and how cytokines can drive the different effector T cells that we have for CD4s. So it's these Treg cells that are able to kind of inhibit autoreactive T cells that are recognizing antigen from the same tissue. So they're able to actually kind of suppress autoreactive T cells. Now, we mentioned natural Tregs a few slides ago. So these are ones that are developing in the thymus through that alternative selection in between the positive and the negative. But we also have induced Tregs. So these are actually induced from naive self-reactive T cells in the periphery. So if you think back to when we were talking about CD4 effector cells and how they can be, we get Tregs as one of that. And if you remember, one of the polarizing cytokines was TGF-beta. So if we have a naive self-reactive T cell that has made it through into the periphery and it starts to recognize antigen and it's being activated, but it's being activated in the presence of TGF-beta, that's going to help it become an induced Treg. So you can see that we've got two Tregs here. We've got the ones from the thymus, so the natural Tregs, and then we've got our induced Tregs in the periphery. But essentially, they play the same role in that they can inhibit other self-reactive T cells, and they do this by secreting IL-10 and TGF-beta. So if you think back to the lecture on cytokines, these were two of the effector cytokines that are produced by Tregs in order to kind of suppress the immune response. So Tregs really important in suppressing that autoimmunity that might be mediated by autoreactive T cells. They do this by the secretion of IL-10 and TGF-beta, but there's two different types. There's the natural ones arising during thymocyte development, but then we have the induced ones that are being induced in the periphery. But they're both playing the key role, and pretty much the same role in tolerance. So if we just look at this whole process, because it's pretty much kind of like a balance in a way. But lymphocytes, they're intrinsically prone to self-reactivity. Okay, we're getting a lot of random gene rearrangements going on during our B cell and T cell development. Now, this is great because it gives us a really broad repertoire. So the more rearrangements we've got going on, the wider repertoire of T cell receptors will be. But also, this gives us a larger potential for autoreactivity. So the more T cell receptors we're making, the more diversity we've got, the greater the potential is that we're going to get some that are going to react with our own tissues, and that is autoreactivity. And that's a big problem, as you mentioned, because that can give us autoimmunity. So therefore, we need to have these tolerance mechanisms in place. And as I mentioned, it's a bit like a seesaw, a bit of a balance, because on one hand, we can have really rigorous tolerance. So that's great, but that gives us a rather slimmer repertoire of lymphocyte receptors. We get a lower risk of autoimmunity, so that's great, but it does increase our susceptibility to infection because we have, that poorer repertoire of lymphocyte receptors. On the other hand, we can have a more permissive tolerance. Now this will give us a very broad repertoire of lymphocyte receptors, which gives us a lower risk of infection, but on the other hand, we have a higher risk of autoimmunity. So you can see it's a little bit of a balancing act as to where we fall within that kind of seesaw. Ideally, what we want is some naive B-cell and T-cell receptors that have a small number of cells for each antigen, but we have a large number of receptors that will give us and confer that protection. So on one hand, we have positive selection, which is ensuring that the receptors that we generate are actually useful. And then we have negative selection, which is reducing that autoreactivity and making sure that we don't get autoimmunity. So it's a little bit of a balancing act just to make sure that we kind of fine-tune our T-cell repertoire so that it is appropriate for our immune system. So just to summarize our tolerance mechanisms, so on the left we have T cells. Remember that they're undergoing central tolerance within the thymus. We're having input from positive selection, negative selection, but also MHC restriction. So remember, MHC restriction is that T cell receptor sort of being able to recognize self-MHC. So we've got that whole thing in play. In terms of the B cells, the central tolerance there is occurring within the bone marrow. Obviously, there's no need for MHC restriction, so we're only getting negative selection. And then for both of these, we have peripheral tolerance within the periphery, and this is what is giving rise to our naive T cell and B cell pool essentially within the body. So if we look at this with a view to autoimmunity, essentially we have these multiple mechanisms or stages, if you want to view them, a little bit like firewalls. So we have our first one, which is our central tolerance. And as I mentioned, none of these are going to be 100% effective, unfortunately. So if the central tolerance fails, we're going to get circulating autoreactive T cells and B cells. So these are essentially slipping through the net. But we have our second firewall, which is peripheral tolerance. But should that fail, then obviously we're going to get autoreactive T cells entering the periphery unchecked, and we're going to result in autoimmunity. So you can ask the question, what changes the threshold for peripheral tolerance? So really, it's kind of quite a complex interaction between our own genes, but also the environment. And if you stick with us next year, sort of in the third year, we will have a look at autoimmunity and tolerance in a little bit more detail. So when it comes to autoimmunity, how is tolerance lost? Well, this is rather an interesting couple of slides from a paper, but what it's showing is this kind of inverse correlation. So you can see we've got the incidence of infectious disease here. So we're looking at a variety of different diseases, but you can see over the last 50, 70 years or so, we've obviously got a decline in these diseases. But at the same time, we're getting this kind of inverse correlation where we're getting an actual increase in autoimmune diseases, but also allergy as well. Now, it's worth stressing, obviously, that correlation is not the same as causation, but there's something going on here. And there's not one reason why tolerance is lost. It's actually quite a complicated picture, and as I mentioned, it's something that we'll kind of look at in the third year module, if that's one that you choose. But self-tolerance, it's obviously very, very important. And we can start to get a breakdown in self-tolerance and the resulting autoimmunity if we have faults in the education of our T cells, but also any faults with tolerance within the thymus or the periphery. That can all lead to that. But actually, studying individuals who have mutations, so for instance, individuals who have mutations in the gene AIR, so obviously we talked about the importance of that. These have actually given us a really good insight into autoimmunity, because these individuals, even though they have just mutations within this particular gene, they present with this widespread autoimmunity. So we can start to see the importance of, let's say, that particular gene in tolerance and autoimmunity. Now, having said that, the majority of autoimmune diseases are not down to one gene. They actually tend to be polygenic, so there's several inputs for a variety of different genes in sort of developing autoimmune diseases. But not only that, we have to factor in the environment as well. So there's various environmental exposures as well as infections, and that can all factor in together with the genetic factors, and this can result in dysregulation and autoimmunity. So to look at these in a little bit more detail and just give you kind of a couple of kind of examples of how this is going on. But in essence, tolerance and autoimmune diseases, it's not fully understood, the precise mechanisms, but we know that we have an input for genetic factors, so anything that kind of alters your immune function, anything that will increase autoantibody production, or perhaps impair your peripheral or central tolerance is going to be leading towards autoimmunity. Also environmental factors, so you can think here bacterial toxins, infections, either drugs as well. Anything that can kind of activate or damage the immune system has a potential kind of input into that system. Also bear in mind molecular mimicry, so if you have viral or bacterial infections that use molecular molecular mimics, these can, it's kind of in the name really, but these can mimic self-antigens, and therefore that can cause us with a big problem. Likewise, anything that affects the immune checkpoints or regulatory T cells is going to have an effect on that and potentially lead us down the route of losing tolerance, but also autoimmunity. So it's really important to understand the mechanisms that underlie these autoimmune diseases, so that we can actually then start to develop a targeted therapy to actually either treat or prevent these conditions. And this is something that I just wanted to highlight because rheumatoid arthritis, so this is something that we're going to talk about in the upcoming lectures in the second half of this module. But rheumatoid arthritis, it's essentially a chronic autoimmune disease, and it's characterized by these really sort of large, painful inflammation within the joints. So you can actually see that in the picture here. But I think the message that I want to kind of drive home is that really by understanding this kind of underlying immunology of these conditions, we can actually start to design treatments or targets that can actually kind of treat or prevent these conditions. So that's a really good reason for actually understanding the immunological basis of these diseases. So there's a Poll Everywhere quiz now with about 8 questions or so if you want to have a go at that. And that will just kind of cover the things that we've gone through today. Okay, it looks like you're starting to get there. So I'll just start to run through these. So the first question, B cell development takes place entirely in the bone marrow. That is, of course, true. Second question, T cell development takes place entirely in the bone marrow. So that's false. So remember, obviously they're arising within the bone marrow, but then you're getting that development in the key stages within the thymus. A developing B cell in the bone marrow binds to self molecules. This B cell can undergo apoptosis or it can undergo receptor editing. So remember, we've got receptor editing or apoptosis. So essentially, it would be receptor editing is kind of having another go at your light chain rearrangement. Okay, so not the heavy chains, but rearranging the light chain genes. A double positive T cell expresses which cell surface markers? So primarily looking for CD8 and CD4. Okay, or both. Yep. So that looks good. Natural Tregs develop in the thymus. That's true. So remember, we're getting the natural Tregs within the thymus, but we're getting the induced ones actually within the periphery. And then this leads on to the next question. Naive T cells can become induced Tregs when they recognize self-antigen in the presence of TGF-beta. So remember, that's the polarizing cytokine for a Treg. And if peripheral tolerance fails, we will get autoimmunity. Okay? All right, that's it. So I think, let me just think. Next week we have the second practical, so there's no lectures next week. So in that case, I will see you guys on Tuesday in the labs for the second practical.